like to speak about viroid-like RNAs that recently has been identified, widespread, and it is a new layer of microbiodiversity that has been identified, but that open a lot of new questions about these infectious or putative infectious RNA elements. So just remember you what viroids are the smallest infectious agents, consist of only a small single-strand circular RNA of 246-430 nucleotides that fold in a compact secondary structure that can be rod-like or branch. They do not, or we don't know that they at the moment are considered non-protein coding RNAs. And this seems to be very simple RNAs. They replicate and move autonomously in the host. They don't need a helper virus for the replication or movement. They are classified into families, Jose Antonio said, nuclear and chloroplastic viroids, and those chloroplastic contained ribosome, cell cleavage ribozymes, the type hammerhead ribozymes that are involved in the rolling circle mechanism of replication. They are subviral pathogen of plants that can induce important disease in the plants. Circularity of some features as circularity of the RNAs, compact that fold in a compact structure containing ribozymes, are properties that are shared by other RNA elements, infectious RNA elements called viroid-like RNAs. Until 2022, only few of them were known and differs from viroids of molecular biological properties, whereas viroids replicate autonomously and are not encapsulated. Viroid-like satellite RNAs that also are infectious agents of plants depends on a helper virus for the infectivity and are encapsulated in their capsid proteins, in the capsid of the virus, the helper virus. And they can contain another type of ribozymes, as you can see here, these hairpin ribozymes. Another type of viral-like RNAs are those belonging to the realm Riboziviria. And one of them, the first identified, was the hepatitis delta virus that infect humans. And also delta virus-like RNAs had been identified in animals, like snakes, birds, fishes, and termites. They also need a dependent helper virus for the infectivity, and they have coding capacity. They code for one protein, that's the delta antigen, and they have a size a little bit longer. They have until 1.7 kb, more or less. And they have contain ribozymes, a particular type of ribozymes called hepatitis delta ribozymes. All these viral-like RNAs replicate through the rolling circle mechanism that Jose Antonio explained, through the symmetric variant that is characterized by the presence of circular RNAs of both polarity strands. There is another kind of viral-like RNAs called retrovirus-like, but they are not infectious, and they have a DNA counterpart in the genome of the organism, where they were found. And these retrovirus-like elements have been found widespread in almost all types of organisms. So until 2022, the viral-like RNAs the infectious viral-like RNAs were found only in plants and animals. Coming back to the hypothesis of the viroids are relics of fossils of a RNA world that is supposed to exist before the work nowadays that is based on cellular work, based on protein and DNA and proteins, that Diener already hypothesized that the virus were fossils from that period. But this hypothesis has a weak point, because if it's true, then you expect to find these viroid-like RNAs also in all the life kingdoms. So another question is, why so few? Until 2022, only more or less 60 species of viroids and viroid-like satellite RNAs were identified. Whereas with the high-throughput sequence technology, thousands of new viruses were discovered. Only few of the viruses were discovered with high-throughput technology, likely because the informatic tools used that are based on homology dependent, or in the homology between the context generated and the sequencing database, fail to find new viroids, viroid-like RNAs, because they don't have identity of sequencing among them. They have identity of structure, but not of sequencing. So probably they escape to this kind of search. In 2023, two independent studies from two groups make high-throughput search looking for new viroid-like RNAs, but with using homology-independent tools. So based on looking for circular RNAs containing ribozymes in both polarity strands. The ribozyme not based on the sequence of the ribosome, but the structure. And these groups were one from published in Cell by Koenig, and the other work was published in Nature Communication in my group, together in collaboration with other groups like as Marcos de la Peña in the CSIC in Valencia, and Artem Abarian from the University of Toronto. So using these homology-independent tools, we identified more than 20,000 different viroid-like RNAs in searching in RNA-seq data, metatranscriptomic data already present in databases. So this result showed that the biodiversity and the number of viroid-like viroids and viroid-like RNA was highly underestimated. This slide resume what we found. The size distribution of the circular RNA genomes with paired ribosomes, with ribosomes in both polarity strands that we found. And it's, so we can see 5 different groups. The most abundant were these with characteristics of the size similar to those of viroids, the small ones. The second group with a size similar to satellites. The third one, the green one, contains the circular RNAs with a size until 2 kilobases, containing the delta and delta-like virus. And we found also circular RNAs as big as almost 5,000 nucleotides, the biggest ones. And when these were analyzed, we found that they correspond to ambivirus and mitovirus that were previously identified as linear RNAs viruses, whereas we found that they could be circular and could contain ribozymes in both polarity strands. And these ambivirus and mitovirus were found infecting fungi. So these results extend the host range to of viroids, viroid-like RNAs also to fungi. Fortunately, we have in our institute a collection of fungi in which these ambivirus were present. So we had the opportunity to experimentally confirm these results, because one of the limits of this search in high throughput sequence data, this bioinformatic result, in silico results, is that in metatranscriptomic data, is that it's very difficult or almost and sometimes impossible to obtain the host, and also the sample where the data was obtained, and you cannot confirm your results. In our case, it was possible, and we confirmed this for the ambivirus. So we confirmed the circularity using the methodology that we already apply to viroids, because we know that circular and linear forms can be separated in denaturing conditions. So whereas in non-denaturing conditions, you have a single band, in northern blot hybridization, in gels, in denaturing conditions, circular and linear form can be separated. So we demonstrate that circular forms of both polarity strands accumulate in the infected fungus. So this is the hallmark of a rolling circle replication. We also demonstrate that the ribozymes that we predict to be in the genome of the ambivirus were active in vitro and also in vivo, because by race we determine the 5' end of the linear forms that coincide with the predicted site of the self-cleavage by the ribozymes that you can see here. The ambivirus also have a coding code contains two ORF, one is polarity strand. ORF A codes for a putative RNA polymerase, RNA-dependent RNA polymerase, but that differs very much from that of RNA viruses, but by structurally correspond to RNA-dependent RNA polymerase, and also contains the motif conserved in the PAN domain of these proteins, and also code in the other polarity strand for ORF P, that is which function is unknown. So the ambivirus are a novel group of infectious agents found in fungi with hybrid features of viroids and RNA-virus. As viroids, they have a circular RNA genome with a compact secondary structure. Here, this present here is rod-like, but they can adopt also, there are other with more branched secondary structure. They have ribozymes in both polarity strands and have identified ribozymes of all types. Hammerhead, hairpin, but also twisted, and also some of them contains hepatitis delta ribozymes. And we propose likely they replicate through a rolling cycle mechanism, But they have also characteristics of the RNA virus, because they code for their own RNA-dependent RNA polymerase. So there will be the first virus, the first RNA elements with a circular RNA, circular genome that is replicated by their own polymerase. So the ICT group ambivirus study group has classified the ambivirus in a new phylum, Ambiviricota, but in the realm Riboviria, in the kingdom Orthonarviriae. So they are considered RNA virus encoding because they encode their own RNA-dependent RNA polymerase. Another interesting thing that we show in this work was that in this experimental confirmation that for one ambivirus, Griffonectia parasitica ambivirus 1, that infected pathogenic fungus Griffonectia parasitica that caused the disease, the chestnut plain, we compare the phenotype of the fungus with Gryphonectia parasitica with and without the ambivirus, and the infection with ambivirus induced hypovirulence in the fungus. So this opened the possibility to use these ambiviruses as possible biocontrol agents. Not only the ambivirus infect fungus, but on another article, very nice, from Wenxi group, they found 3 viroid-like infectious agents in the fungus Potriosphaera dotidea in 3 different isolates. This fungus is plant pathogenic, causes important disease in trees and wine, and this group confirm experimentally that they are circular. They identify these circular RNAs with size from 157 to 450 that are circular. They adopt as a compact conformation, branches that remember very much the chloroplastic viroids, replicate autonomously in the nucleus. They convert the autonomous replication, probably via rolling cycle mechanism, and they prove that they are infectious. They generate infectious clones, these infectious transcripts, dimeric transcript head to tail, as we did with viroids, and they infect a fungus without that. They transfect this RNA, and they obtain in the progeny for, I think, until 6 or 7 progenies, they can detect these circular RNAs. And also in this case, the infection with Potriosphaera dotidea circular RNAs induce pathogenic effects in the host, decreasing the growth rate, attenuate also the virulence, and increase the decrease of tolerance to osmotic and oxidative stress. So these authors propose to call these infectious RNA elements like mycoviroids, but they couldn't find any ribozyme in the sequence, any known ribozyme structure in these RNAs. Whatever, one dimeric form of these RNAs was able to self-cleavage in vitro. So probably an unknown ribozyme structure is present in these RNAs. So this remains an open question. And last year, another study, a high-throughput search in databases based in a non-homology methodology bioinformatic tool, identified a novel type of viroid-like RNAs, widespread in human microbiome, and now this year, a few months ago, also another group published that this new type of viroid-like RNAs was found in the oceans, in the prokaryotic fraction. So, and this new type of viroid-like RNAs they call obelisk. The obelisk are formed by have a predicted circular RNA genome of about 1 kilobases, adopt a almost perfect rod-like conformation. They have coding properties. They code for a novel protein, oblin, all of them, that is that show a specific tertiary structure, predicted, of course, and that is conserved in all of them, which function is unknown. And some of them, only a small group of them, contains hammerhead ribodes in both polarity strands. Only one host was confirmed for this obelisk, and they found these 30,000 of these types of obelisks widespread in human microbiome. And in the oceans, if you see here in the title of the article, they exceed the abundance of RNA viruses in the prokaryotic fraction of the ocean. So it's a huge amount of this kind of putative viroid-like RNAs. So they are widespread, but only in one case, in one was the host was proved, was confirmed in one bacteria, oral human bacteria, Streptococcus sanguinis. So the impact in the human health that these new viral-like RNAs can have, and the ecological impact that this viral-like can have in the oceans is completely unknown. So all this bioinformatic, all this identification by bioinformatic tools open a lot of questions. First of all, and to answer these questions, we need, first of all, to identify the host of these viral-like RNAs, because they were found in metatranscriptomic data from different ecological niches, different sources. But we need to know the host to follow all these confirmation, experimental confirmation that are necessary to go ahead. And then we need to answer if they are effective, the infectivity, as the title of the talk say. To prove the infectivity, we have to construct infective clones, infected transcripts, and to obtain isogenic lines, for example, in the fungus, infected with the viroid or viroid-like and not infected, to prove the infectivity and also to the impact and the biological effect in the host. For example, in the fungus, where if the infected fungus is more pathogenic, is in the cases of beneficial fungus for the plant, if increase this beneficial effect, or even if it's latent. And there are also another basic questions about their replication. Which polymerase is involved in the replication? Is the RNA pol 2, as in the viroids, the site of replication, and also if the mechanism is the rolling cycle. And some of them we saw that they contain, or that they could code for proteins, which the expression of the strategy to express these proteins is unknown, and also the function of the putative encoded proteins that are completely known. And another is not a problem, is an issue. This huge amount of new viral-like RNAs must be classified. And what we can say that is a viroid, what we can consider this a viroid, that must be defined. And recently in PLOS Pathogen, there has been published it's called PERMS by Bizarro, given some consideration to what things that we have to take account to consider before proposing that a circular RNA is a viroid, a classical viroid. So he said that must not code for proteins, presence of functional structures, that's it, must have a compact secondary structure, branch or rod-like, must replicate autonomously without the presence of a helper virus. So we must be sure that when we infect, there is no virus present in the host. And we have to prove the infectivity, the transmissibility, and if they are pathogenic or not. So let's say the Koch postulates that sometimes we forget about them. And in the case of Absum viroide, the presence of ribozyme.